Hello, and welcome to Virtual Investor Conferences. On behalf of OTC Markets, we are very pleased you've joined us for the Life Sciences Investor Forum. Our next presentation of the day is from Thiogenesis Therapeutics. Please note you may submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings by clicking Book a Meeting in the top toolbar. At this point, I am very pleased to welcome Brooke Riggins, Director and Chief Financial Officer of Thiogenesis Therapeutics, which trades on the OTCQX Best Market under the symbol TTIPF and on TSXV under the symbol TTI. Welcome, Brooke.
Thank you, Lily, and welcome everybody. So Thiogenesis is a biotech company, clinical stage biotech company. We are starting off with a orphan platform, which is orphan drugs under 200,000, which allows smaller, faster clinical trials. And we have a rapid clinical path based on the fact that our lead compound, TTI 0102, is a prodrug and based on a previously approved compound. I will go forward. The forward-looking statement, we have a few in the presentation, and you feel free to go back and look at it later if you need more time. Thiogenesis has a very experienced leadership team in the orphan drug space. Our chairman is Chris Starr. He was a co-founder of BioMarin, where he helped lead bringing 3 products to market and left that company as it was approaching $4 billion in market cap. He worked as a co-founder and CEO of Raptor Pharmaceutical, which went public at 20 million in market cap and was sold for 800 million. And he's also the co-executive chairman of Monopar, which recently raised 160 million and has a market cap of about 565 million. Patrice Rieu is our founder and CEO. He's got an extensive career in mitochondrial disease, mitochondrial metabolism. He also was the head of ClinReg at Raptor as they got the lead product Persisby approved for one indication called cystinosis. And why I have those both highlighted in purple for this slide is that our prodrug is based on the same active ingredient, cysteamine, that was in the Raptor drug, which gives us extensive experience in dealing with cysteamine and all the approach to regulatory. As I mentioned, we're a clinical stage biotech, which means we've been tested in humans. It's a thiol-based technology, which is what cysteamine is, which means it has an active sulfur hydrogen component. You can think of sulfur as being the big challenge there. It's bright yellow, it smells like rotten eggs, it's smelly, and it's very difficult to digest. And we are addressing these applications for high-need pediatric and orphan diseases. Cysteamine, which we're talking about, has been studied across several diseases for several decades, and its mechanism of action and biology is extremely well understood. It's been approved for one indication, cystinosis, that was is a generic called Cystagon, and then later Persisby for Raptor. However, it has promise in much bigger indications, and those are all based around mitochondrial dysfunction and oxidative stress of the mitochondria. The mitochondria, just as a preamble, is your energy source in your cells. It takes glucose and oxygen and creates energy, which is necessary to drive the metabolism of your cell. And when it gets less efficient or diseased, it throws off free radicals and produces less energy and creates chronic and longer-term disease. We have active clinical programs, a phase 2 in Malage, which we did an interim analysis about 2 or 3 weeks ago, and we expect final data at the end of January. We have an IND cleared in the United States with a leading children's hospital to do Lee syndrome. Both of those are inherited mitochondrial diseases. We've had communications with the EMA to do a pediatric MASH study, that's fatty liver for kids, and they've given us guidance on how to file an IMPD to start a phase 2 trial, which we anticipate filing sometime next year. And we're also just recently announced that we're going to be going into a phase 3 study in cystinosis. Cystinosis is the one indication that has been previously approved, and that was with Persisby, and we will look to have a superior drug in a phase 3 trial next year. Our valuation right now is roughly US 25 million, Canadian equivalent of about 35 million. which is significantly lower than the comps, that particularly the ones you'll find in the United States, which range from 100 million US to about 450 million US. Our investment highlights, just to start, is we begin with having a cysteamine-based drug that's been used in thousands of patients, dozens of studies, actually more than dozens of studies, hundreds of studies, and it has a very strong rationale built on supplying glutathione, taurine, and the antioxidant profile. It's a next generation cysteamine prodrug, which improves GI tolerability. As I mentioned before, sulfur is very hard to digest and can cause nausea, indigestion, vomiting, etc. And what we've created with our prodrug is a new chemical entity that uses the metabolism to slowly release the cysteamine and not spike, so that you've really limited the side effects. Very important to improve adherence to taking the drug, and also it improves dosing flexibility, so we can give more drug, which is really important for some of the bigger indications. We rely on the 505(b)(2) regulatory pathway, which helps us a lot because it allows us to skip some of the safety and phase 1 work that new drug development entails, and that can save you 4 or 5 years and several $1 million, and allows you to go directly into phase 2 efficacy studies. Our 3 leading indications that we've talked to with FDA and EMA, MALASH, Lee syndrome, and pediatric MASH, have all received positive reviews and allowed us to start a MALASH study, have an IND cleared for Lee syndrome, and have guidance to clear a IMPD for MASH. And we also already know the pathway to getting a phase 3 started in cystinosis, having already had a drug approved in cystinosis. What's really important about our model is because it's based on a previously approved drug, which we have a lot of experience with, we can drive proof of concept and value with really small, less expensive, faster clinical trials. We try to design them between 10 and 20 patients so that we can get them done in the range of around under $5 million and within about 18 months. Our lead compound, TTI-0102, it's what we call an asymmetric disulfide, which means it has that active compound, cysteamine, and we've synthesized it with another thiol, which is called pantothane. And what creates the 2 cysteamine molecules and the pantothenic acid is the way it gets metabolized. So as a prodrug, it's not active, and then once you swallow it, the metabolism starts to pull apart as it gets reduced. The first cysteamine compound, which is very slow and without side effects because of the slow nature of the metabolism. And then the second compound, pantothine, goes into your small intestine, where it actually recreates another cysteamine compound with an enzyme in your small intestine. And it also creates pantothenic acid, which is really important for metabolism. This controlled process of separating and then recreating is what we call a metabolic gating system, and that allows the drug to be produced without a peak, which reduces the GI gastrointestinal side effects and allows us to give more drug and make it last longer. That gives us the extended PK by giving more drug, which can theoretically allow us to give the drug once a day. Also, without having the side effects, we know with Prisisby that there's vomiting and nausea involved. Twice a day, we have a better chance of having patients comply with the drug scheduling, and also have it be used for chronic long-term use. One of the key elements of the TTI-0102 is to boost intracellular cysteine, which is a amino acid. And if you look to the left, you'll see what we're addressing with the mitochondria, the powerhouse of your cell. On the left is a young mitochondria, healthy, which takes glucose and oxygen and creates ATP energy, which is the energy required for your cell's metabolism. And to the right is either an older one or a diseased dysfunctional mitochondria, which takes that glucose and makes energy, but it also makes a lot of free radicals. So you get less energy, and B, you get these toxic free radicals, which will then cause cell death and other issues with lipids and DNA. And so it's the reason that intracellular cysteine is so important is because that's a precursor to glutathione, the master antioxidant, which will neutralize those free radicals, and also for taurine, which will help with neuronal stability and reduce seizures and other issues that you get with mitochondrial dysfunction. What's really important about this slide is that being able to be the leader in creating intracellular cysteine is that this mitochondrial dysfunction is something that's across chronic disease. We're starting off with inherited mitochondrial disease as a proof of concept because it's quite the most obvious there that if you have a mutation where your mitochondria doesn't function properly, you can address that and there's no good drugs for those diseases. But it also happens in chronic disease, things like Alzheimer's, Parkinson's, diabetes, et cetera. And so it creates a really strong platform for future drug development. One of the key features of our drug, as we mentioned, was that you reduce side effects and you can potentially give a lot more drug and have it last once a day. We did a phase 1 study in Australia with 12 healthy volunteers where we increased the drug up to 4 times the therapeutic dosage of the generic version of cysteamine. And as you can see with that red line above, it was released slowly and surely. That's the green line in the chart. But it didn't hit that full spike, which you would get associated with the side effects. And then the dotted line that you can see in the middle over the 0.1 is then the amount you need in the system to be therapeutic. And you can see that in this, when you give 4x the amount of drug to a healthy adult, that it would last 24 hours. These patients did not report any significant side effects. Some of the nurses noticed that there was a bit of body odor related to the rotten egg smell, but it wasn't coming from the patients. So that is superior to what we've seen in previous cysteamine-based drugs. One of the pieces of the puzzle that we've just added is that we originally were looking at all new indications, but we also realized that the TTI 0102 has a really great profile for cystinosis, which is the one indication that's been previously approved for. And so that's about 1,000 patients in the U.S. It's a roughly 300 million addressable market in the U.S., probably 400 million worldwide. And what cystinosis is if you look at to the left, that's what's called the lysosome. That's your recycling bin inside your cells that gobbles up sort of foreign materials, dying materials, proteins, et cetera, and it recycles them either for waste or to be recycled. And what happens in the case of cystinosis is there's a compound called cysteine, which is unable to leave the lysosome. And when it can't leave the lysosome, it can turn into crystals. And those crystals will accumulate in your kidney, your eyes, and your organs, and they're very toxic. And so what happens, this is a mutation, so it's a childhood disease, usually gets diagnosed in the 2 or 3 years old, is that you would have to have without treatment, you'd have to have a kidney transplant and your mortality is limited. But with treatment, lifelong treatment of cysteamine-based drugs, you can live into your 40s and 50s and potentially save kidneys and other organs. What happened originally I think in the 90s was they used the generic version, which was 4 times a day with extreme side effects, GI side effects. And in around 2013, I think it was, Raptor got Precision approved, which was a coating around the generic version of the drug, which allowed you to take the drug twice a day, once before bed and once in the morning, which was a big improvement. And we had some anticipation that it would reduce the side effects. It did a bit, but not significantly. And now we're able to use, we would be able to use TTI 0102, not twice a day, but once a day, and eliminate the side effects. And that's the big upside to the TTI 0102. We think we can do that as a non-inferiority trial where we just test it versus the historic levels of Precision and see that it's just as good. And then the marketing based on once a day and low side effects should be a big advantage. And then in regard to why I've circled the cysteine down below, as I mentioned before, cysteine, when you have chronic disease or mitochondrial disease, cysteine would be low because it's all being used to battle the free radicals. And so by tearing apart the cystine and exiting once piggybacked with cysteamine, but releasing that cysteine, you get this amazing amount of cysteine available to make glutathione and taurine, which is part of the second part of the platform, which is able to battle mitochondrial dysfunction. We announced in June, speaking of mitochondrial dysfunction, that we would we got an IND cleared in collaboration with one of the very leading children's hospitals in the United States, one that has collaborated with and helped develop lots of technologies that have gone on to successful exits. And we got the IND cleared. Leigh syndrome is probably the most difficult of the inherited mitochondrial diseases to treat. There's about 300 patients, we think. And you get a double mutation, and the kids just don't develop. They don't have energy to develop. They have trouble sitting up to breastfeed. Their life expectancy, 50%, could pass away before 3 years of age. And they have really low cysteine, glutathione, and taurine. Oftentimes they treat the symptoms, and they can use supplements, something like NAC, which is a supplement that does sort of what cysteamine does, but at way lower levels. And with this FDA cleared IND, we ordered the drug and are ready to start a phase 2A study in the first quarter of next year. The first stage is 9 patients. They'll be adolescents. It'll be 6 on drug and 3 on placebo. We'll be looking at all the biomarkers, cysteine, glutathione, taurine. We'll also be looking at some fatigue severity scale and other measures of quality of life. We would anticipate, if we start that in Q1, we'd anticipate having that stage 1 data in Q3, maybe September or October. We started last year a proof of concept study in MALASH in Europe, primarily at Radboud University in the Netherlands, but also one site in France. And MALASH is not quite as severe as Leigh syndrome, single mutation. Patients are diagnosed usually 5 or 6 years old. It also has oxidative stress, low glutathione, taurine, et cetera. And in that study, we gave the maximum dosage to the whole range of patients. Some of the patients were not able to tolerate the drug. They were the small ones, below 50 kilograms. So we've used that information to recalibrate our dosing regimen in future trials so that we'll give a smaller dose to smaller kids. It'll be sort of 2 uniform doses, and then the full dose for big kids and adults. And we did an interim analysis where we saw really nice biomarker data. We saw evidence of small early evidence of efficacy and some biomarker improvements and dosing insights. So we are looking forward to doing some form of conference either late January or early February, and giving the final readout on the MALASH study. Just so I can give you an idea of the market size for Leigh syndrome, MALASH, et cetera, is we're using those as proof of concepts for that whole area of inherited mitochondrial disease. There are roughly 75 to 90,000 patients in the US with inherited mitochondrial disease, and there's about a dozen of them. And so we would anticipate, if it works in Leigh syndrome and MALASH, that we would go after far bigger market than just these 2 indications. This is a bigger footprint for us. This is our large indication, which we've had that feedback from EMA that we can go into pediatrics, which a lot of the GLP-1s and fatty liver companies, they haven't resolved their safety issues yet to be comfortable going into children. And we had discussions, because we're already a pediatric company and in kids with our drug and with the previous version of cysteamine. And they gave us guidance on how to file to go into pediatric MASH in Europe in a phase 2. We'll probably file that around the second quarter of next year. What's really interesting is what we did at Raptor. There was a study, an NIH study on fatty liver in kids, 169 patients. And originally, it didn't show significance for pediatric, the fat in the liver for the pediatrics. However, we used a really low dose because of the side effects. But when we did our retrospective analysis, the ones that were under 60 kilograms had a very nice p-value, 0.001 of reduction in fat in the liver, whereas the ones that were bigger weren't, and they skewed the data. Now we can increase the dosing by anywhere from 2 to 4x we feel we can go after both the smaller and the larger patients. What's really important there is if you can get the mitochondria more functional, that it's better able to metabolize the fat in the liver, which we saw in the NIH study. But there are secondary mechanisms involved with that second compound that we attached to the TTI 0102, which also gives some benefits that we think will reduce fibrosis, which is really a holy grail for fatty liver disease. And so we think that the IMPD and a proof of concept study there would be very valuable to the company, and that would be subject to funding in the second half of the year. Upcoming milestones for the company. We have the cystinosis. We look to file the IND in this first half, middle of the year. We have to have the final version of the drug, not the experimental version, to do that. And then once we've got that filed and cleared, we'll be able to model out, the size of the trial necessary to approach that $300 million market. Lee syndrome, we anticipate dosing in Q1 of next year, and we'll probably have more guidance on who we're collaborating with and design of the program at that stage. MALASH, we'll have phase 2 final data at a conference at the end of January, early February. And then we also expect to have that IMPD cleared for pediatric MASH probably in the second quarter of next year. That in itself will probably get some attention in regard to talking to some of the players in that space, because everybody's looking for non-GLP-1 approaches to metabolic disease and fatty liver disease. This is our pipeline. I put stripes on the phase 2 because we probably get to skip phase 2 and go straight into phase 3 for the cystinosis, phase 2A for Lee syndrome, phase 2 for MALASH. And then this is our cap chart. 51 million shares outstanding, 56.9 fully diluted. Those are all employee and management incentive options. We've done 4 rounds of funding, all of them straight equity without any investor warrants attached. Insiders control 16.5 million shares, about 30% of the company. Market cap right now is about 33 million Canadian, 25 million US, and the comps that we see for orphan drugs through fatty liver, lysosomal storage disease, et cetera, range between 100 million US and 450 million US. Our cash position at 9/30 was 3.3 million Canadian. Okay. That's the formal presentation, and I see there's a few questions. I can start having a look at these. I think I lost one of them. So I've got one question about how much capital we need for all 4 indications through 2026. I would suggest that the MALASH is going to stop after, by the end of this year. We'll need to finance the Lee syndrome and the cystinosis. We'll have to order the drug where there's a lag, and the fatty liver is a second half of the year story also. So our minimal capital requirement for next year would be $5 million US, which, as we said at the beginning, we have a very good use of proceeds and capital management program. How far does our cash flow run right now? It runs to the end of May. We will anticipate raising capital in the first quarter of next year. Another question here in regard to our MALASH phase 2 data package. That was the way to look at the phase 2 MALASH study was a couple of things, and I can't give too much information and stale a potential publication or abstract, but we did get the positive biomarkers we were looking for. We had other proof of concept issues, things resolved there, and great information in regard to our dosing, which we're applying to the Lee syndrome study. And so we will probably combine the MALASH data and the Lee syndrome data in a future phase 3 study. So we will not probably initiate the phase 3 immediately, and we'll look to combine those areas in under one sort of umbrella going forward. How would we, because cysteamine is approved as a generic, and also with Precision, how will we differentiate it commercially and pricing power wise? I would suggest that one, we would probably be equivalent in pricing in regard to Precision, first of all. Secondly, the differentiation will largely be like as we said before, if we can potentially dose it once a day. You might be twice for the smaller kids, but at a certain point, they'll be able to tolerate once a day, which is a big differentiator. The side effect profile would be much improved. You will have far less nauseousness, far less vomiting, and there's some issues with fibrosis that comes with the enteric coating. And so the commercial profile of TTI-1 would be significantly better in regard to cystinosis, and we probably won't face competition from the other cysteamine products in the new indications around the mitochondria or fatty liver. Considering an uplist in the U.S. One of the issues that I think that is really important in regard to our valuation is we do have a U.S. very NASDAQ savvy board of directors and experience in the leadership level. And we listed in Canada, largely myself and another director were Canadian, had access to capital, and the biotech markets two and a half years ago weren't that open for small companies. And so, yes, ultimately we do want to uplist in the U.S. And one of our big, we have done reasonably successful financings without broker warrants or like investor warrants attached to them. And so our feeling is that our next level set for valuation will be increased exposure into the US market. This is one of the first discussions I've had directly with US investors, but we also intend on going to see institutions, investment banks, analysts, et cetera, largely driven off of the MALASH completion of the phase 2, the starting of the Lee syndrome in phase 2, and clearing the phase 3 IND for cystinosis. I think that's it for the questions. If anybody wants more information or to follow up, please reach out at info@thiogenesis.com. I receive all of those emails and I would be happy to respond and follow up.